Everyone, welcome to today's webinar. So today I will introduce one of the new features in the Pix11. It's our DeepNovo Peptidome workflow. So I will introduce some new features in Pix11 first. And then I will introduce the features for the DeepNovo Peptidome workflow. Later, I will show a demo project. We use the Immunopeptidome dataset to show the sensitivity and accuracy of our workflow. And later, I will show some research highlights from our Pix users, those who use the Pix software to do something like the peptidome or immunopeptidomes. And finally, we will show our software to show you some functions and how to use our software. So let's get started. So first thing is that now we launched the Pix11 family. It includes the Pix Studio 11 and the Pix Online 11. So basically, the Pix Studio is a complete software it's a standalone software. So it's designed for the single use user. And the Pix Online is a web-based software, so it's designed for the large cohort data study, and they use the web server to help the multiple users. So if you are interested in our software, you can visit this link to our Pix11 to find more information. And for the new features in the Pix11, we will have several webinars in following weeks. So if you are interested, you can also register in this website to follow our new features. Okay, so now let's start from the DeepNovo Peptidome workflow. So the first thing is that this workflow is a built-in workflow in both Pix Studio and the Pix Online. And it's designed for the peptidome search. So let's start from the peptidome. When we say peptidome, we assume that the peptides is generated from the protein by the proteolysis. So usually when we do the database search, we will do the unspecific search. So the most representative example is the immunopeptidome. Just like the picture showed here from a paper in 2020, this year. At first, the protein is proteolyzed in vivo, and then it becomes the peptide, and then it's presented by the MHC molecule on the surface of the cell, and then the T cell can recognize that. So why we are so interested in the immunopeptidome is that the immunopeptidome can link to a very hot research area now. It's the cancer immunotherapy. So we can do the CAR T treatment, or we can develop the mRNA vaccine based on the immunopeptides. And I want to point out here, our workflow is designed for the peptidome. That means not only the immunopeptidome, but also some peptidomes, something like from the food, from the plant, from the dairy, or even from the marine animals. So it's a peptidome workflow. It's not just for the immunopeptidome. And then here are some new features of our peptidome workflow. First thing is that it's a built-in workflow. That means you select this workflow and set some parameters, and then click Start. It's very easy for the users. Second thing is that it's designed for the peptidome research. That means we optimize some search parameters for the peptidome, not what like we used in the general proteomics. So I think there are two key features. The first thing is that we combine the different search strategies. We use unspecific database search and the BSI Software spider search and the DeepNovo search. And the second thing is that all the result is controlled by FDR. That means whatever the peptides from, it's from the DB or from the DeepNovo, it's all controlled by the FDR. And then to increase the sensitivity and accuracy, we use the deep learning predictors to predict the MS spectra to help user to filter or to review the results. And let's start from the search strategies. So usually, if you are familiar with the peaks, we know that the peaks is start from the de novo sequence. That's because de novo sequences are not according when peptides when the protein faster here. So we can start from here, and then if I got the protein sequence, I got the FASTA file. I can do the database search. Finally, when I get the de novo result and the DB result, we sometimes will find some MS2 spectras have good de novo result, but it cannot be matched in the FASTA file. At this time, we can do the spider search to find if this peptide is a mutation or is there some sequencing error. And this is our workflow for the DeepNovo Peptidome workflow. At first, we have the raw data. Then we will do the DB search or the DeepNovo search. Here, when I say the DB search, it's also included the spider search. Then we will get the result. We divide the result into two parts. The first part is the canonical peptide. It's usually we think if the peptide is from the DB result, it belongs to the canonical peptide. And then it's the non-canonical peptide. Usually it's from the spider search or the DeepNovo search. Then we combine these three peptides to generate a new FASTA file and do the DB search again. In this round of the DB search, we control the FDR to get the final result. And then based on these results, we do the binding affinity or the immunogenicity predictions. And now we talk about the peptidome. Now let's talk about the DeepNovo. Why we need the DeepNovo here? We know that when we do the immunopeptidomes, the most interesting we want is the neoantigens. That means it's a new one. So just like what described in this paper, we will find a different source of the neoantigen source. And what's more, there's a more latest paper report that the non-canonical peptides made from the splice is from the splice. So all these things should be. We can say that the most important thing is that the non-canonical peptides, their sequence may be not included in the protein FASTA file. So that means if I want to do the DB search, but if the sequence is not in the FASTA file, how can I do the search? So now usually we will have an alternative. We do the proteogenomics search. That means for each sample, we can use the mRNA sequence result for each sample. And then we search against this personal FASTA file. But there is one issue here. Usually the size of the personal FASTA is very large. So the search space is large. In this situation, the FDR estimation will be an issue. So in this time, we need the DeepNovo here. So in simple, the DeepNovo is a deep learning based de novo sequencing technique developed by our BSI. And in here, we do not require the protein FASTA file. We just deduce the sequence from the MS2 spectra directly. And then the most question we meet is that, yes, you can do the DeepNovo, but how can you evaluate your result? For those who are familiar with the PICS version, you know that we have a score called the ALC score. It's the amino acid local confidence score. If the score is higher, that means this match, this PSM is very good. So that means this may be a right match. And now we have the deep learning. So we add more attributes or more attributes to help the user to see if this is a good PSM. So the first basic one is we have the classical PSM viewer. So in this viewer, we can see that D and YN, and we can, based on the distance between two peaks, to see if this PSM is right. And now we have the deep learning prediction. We can predict the MS2 spectra based on the input sequence. So besides this viewer, we have the mirror plot here. So on the top of the figure is the measured spectra, and on the bottom of the spectra is the predict one. So we can compare the measured spectra to predict spectra based on the ion type, and the most importantly is the relative intensity of each peak. And what's more, since we use LC-mass, so we have the retention time. So here we also predict the retention time for each peptide. So now in the peptide table, we have two new columns. The first one is the RT, it's to measure the retention time. And then we have the delta RT, it's the difference between the measured one and the predict one. So based on this value, we hope that if we assume that if it's a good match, then the retention delta RT should be very small. And in the result summary page, in the summary page of the result, we will give you the statistic plot, something like the histogram and the regression line, to help the user to see if this data set is good and if the match is good. So for take this sample as example, most of the peptides in this sample is delta RT is less than 10 minutes. And whatever it's from, whether it's from DB or from DeepNovo or from the homology, all these peptides fit to the regression line. So we can say that it's a good match. The PSM in this search is good. What's more, if the input data is from the team's top, we can also predict the CCS value. So that means we predict besides the MS2 spectra and the delta RT, we also give you a column the delta CCS to see if it's a good match. And now we can take an example. So this is a match. I think we can give you 5 seconds to see if this is a good match. Okay, so for me, this PSM sounds very interesting. So the first thing, if we just consider the ion type, that means we see the Y2, Y3 here. Looks like all the ions are matched to the predict. But if we also consider the relative intensity, so then we will find that for some peak we predict here is missed in the measured spectra. So it's hard to see or hard to judge if this is a good match or bad match. So in this time, we need more attributes to help us. So the first one here, we have a column we call the CAA. That means the confidence amino acid threshold. We will explain more detail in the next slide. And then we have the first column, the delta RT we see. And also we have the MS2 correlation. There's the similarity between the measured spectra and the predict spectra. So we see here, the first is the delta RT. If you remember in the last slides, for this data set, most of our peptides, their delta RT should be less than 10 minutes. So obviously, this is the outlier. So we can see the delta RT is not good. And then we check the MS2 correlation. It's also very low, just 0.39. So based on this, we can say, oh, this is not a good PSM. We can filter it. So in Pix11, we add more confidence related attributes to help the user to validate their results. And then let's explain more about the CA values. So the first question is that some user will ask why which amino acid have the different color. So what are the different color mean here? So we can, if we will click on the icon here, you can see by default we will say if the CA is less, if the CA threshold is less than 1, then the peptide is the color is black. If it's larger than 5, then it's red. So usually we set the value is 2, then we set here. And what is the CA confidence? What is the CA score? Well, here is the explanation. For the CA, we say this is a support ion. So there are two standards. The first thing that if we want to say this ion is a support ion, so we will check its intensity. The intensity here should be higher than the threshold I set. For example, I set the threshold is 2, and the intensity for this peak is just 1.8. So it's lower than the threshold I set. So this ion will be not considered as a support ion to calculate the CAA. This is the first thing. So for those who are familiar with MS spectra, you can consider this value just something like the signal to noise, the SN of the peak. The other thing is that if it wants to be a support ion, it should have the fragment ion in either side. That means if this peak is a support ion, it should have either B ion or either have the Y ions. So if one amino acid could be counted as a confident amino acid, it have two standards. The first thing is that the peak intensity should be higher than what we set, and then for these ions, its fragment ion should have fragment ion in either side. That means you must have the B ion or have the Y ion, and then we say, okay, this amino acid is a confident amino acid, and then for one peptide, we count the ratio of the confident amino acid to get the final CA values. Okay, then now we can have an example to show how to use the CA value here. So in here, I just set the default value from 1 to 2. So here, for these peptides, you can see the difference here. At first, it reports the sequence, the N-terminal sequence is DA, and then the other sequence. And now, if I set the CA threshold is 2, you can find that now that in the N-terminal, the sequence disappeared. It's just give you a number. We call this is a must tag. So why it become a must tag? Then we can check the first thing is we can check the CA value or the value here. We can find that besides the other amino acids in these peptides, the N-terminal here, they tell me that the CA is 0. But why? Then we can check the raw spectra here. We found that in this spectra, we just find the B2 ion here. So that means we only know these peptides have N-terminal that have D and A. But we don't know it's BA or AD since we didn't find the B1 ion here. And in the Immunopeptidome, this is very common if you use the CID or sometimes HCD. It's very common that we will lose the N-terminal ions here. And in this time, we can just say that we cannot sure the sequence, and this time we need to other validation. So that means if I have the motif informations, then I can check this sequence with the motif. So for this data set, since we know that it's from the human, it's from the HLA 8601. So we know the motif should be like here. So we know we can compare that. We know the first sequence should be D and then it's A. So based on these two information, we can say that, okay, then the DA may be the right answers. This is the way how we use the DA values. And in the peptide tables, we have the peptide field here. If you click the peptide tables, you can have the peptide filter. So usually we will recommend the user to filter peptides based on the CAA, and then based on the MS2 correlation, and based on the delta RT. And especially if you want to check the quality of the DeepNovo peptides. Then we can use all these features to show our demo project. Our demo project is built from the dataset from this paper in 2021 from the research from the research of the Switzerland. So in their paper, they build the EDA dataset and the DIA dataset from the human cell line and process them with the different software. And we pick up one of the dataset. We pick up the cell line from the RA957 to process the analyze in our workflow. So before the analyze, so if I was a user, I want to ask 3 questions. The first thing is that if Pix11 content workflow can process the DDA and the DIA data for the immunopeptidome. And the second question is that if you can process, how about the sensitivity? If you can find more peptides and you can cover more, that means you can cover more peptides than reported. And then is the accuracy. Usually we will check the peptide length, motif, and the binding affinities ratios to see if your result is good. So for the DDA data, we process it in our DeepNovo Peptidome workflow, use the comparable parameters. And then this is the result. Usually we have a summary table in our result. So one thing I should point out that based on this FDR curve, I set the more strict the FDR. We use the 0.1%, not the 1% reported in the paper. Since based on the FDR curve, cut here is more reasonable. And this is the result summary. So we will have a table. So based on our workflow, we can find around 13,000 peptides from the DD, the spider search, and the DeepNovo search. And here is the MS shift figure. And here is the rotation time, delta RT figures, delta RT shift. So based on this figure on the population level, we can say that the data set is good. Our analysis is good since they are both of they are the normal distribution and their cutoff is less than what we said. Then we compare the peptide number. So in the paper, it reports around 50,000 peptides from three replicates. And in our result, we report almost double of the number. And then the following question is that you report double, but can you cover the peptides which report in the paper? So we compare the peptide sequence report in the paper and in the Pix11. So, and we found that the majority, nearly 93% of the peptide reported in the paper can be covered by the Pix11. So we can say that the sensitivity is good, and also it can cover most of the report sequence. And here is one reminder. So since this data is from the DDA, so I also want to see that if our workflow is reproducible, it's stable. So I used the other new features in Pix11, so our QC to assess this data. So we can find that although it's just the DDA replicate, it's also the data set is also highly reproducible. So we can see the ID number, and around 91% of the peptides could be found in all replicates. Then, if you are interested in this QC style results, please for our next webinar, we will introduce our QC functions in Pix11. Then next, we will check three attributes of our results. We will check the long distributions, the motifs, and the binding affinities of our results. So this is the long distribution. In the left is the report in the paper, and in the right is our result. So the long distribution is similar. It's okay. And then we compare the motif. This is report in the paper, and this is report in the Pix11. And we check the motif and motif. They are the same. Then we use the same algorithm to predict the binding. So we find that around 92% of our report peptides are binders. These are very high. So based on these things, we can say based on the long distribution, motifs, and the binding affinities, we can say our workflow is a sensitive workflow and also is an accuracy workflow for the DDA data. Then we convert to the DIA data. So we not compare our results with the original paper, but we compared with a paper in last year. It's from the Dr. Matthias Mann's group from Germany. They use their deep learning models to reanalyze the DIA data in the DIA data. And also in last year, so our BSI also have a paper to report our streamlined DIA workflow. So in the DIA workflow, we use the special library search, DB search, and the DeepNovo search. So to offer a complete workflow to analyze the DIA data. So we also process the DIA data in this workflow to see if the result is comparable. And we use the comparable search parameters, and we all use the human reviewed sequence to do the DIA search. This is the summary of the result. And based on the mass shift or the retention time, we see that our result is stable, and the delta RT, the RT regression is very good in this data set. And here we compare the peptide sequence, use the same filter. That means we just compare the peptides the length between 8 to 14. And we report a close number to the paper. Both of our sequence report around 36,000 peptides. And we also compare the overlap between two algorithms. So the first thing is that around 64 of the peptides reported by Pix11 can be overlapped by the paper from the Matthias Mann group. And for each algorithm, around 26% of the peptides are unique in each of the algorithms. So for two algorithms, this is very common since each algorithm may have its performance sequence, and our Pix11 and the alpha cap depth may have some different performance for the sequence. So the result is still reasonable. Then we also compare some attributes from the long distributions, okay. And then also we compare the motif. So we report the same motif. And then we use our peptide to predict the binding affinities. Well, in this case, our over 85% of the peptides we reported are binders. So it's the same when we check the longs, motifs, and the bindings. We can say it's the workflow for the BI is also sensitivity and accuracy. And then before we go to the demo show, I want to show some research highlights from our PICS users. The first one is that the research from Canada, from the University of Montreal, they report that they use the iPSC sample and they report 6 immunogenetic peptides. These peptides they found, half of their peptides are found by the personalized FASTA file. So we use the peptidome workflow and just use the reviewed human canonical FASTA file from Immunopeptidomics. We want to see that if our peptidome workflow can find these immunogenetic peptides only found in the personal FASTA file. So here you can see that. So for this sequence, they are canonic. They are in the reviewed database. We can find them. And also, if for these 3 peptides, their sequence are only in the personal FASTA file, not in the canonical FASTA file, we can also find them by our DeepNovo. So this shows our accuracy of the workflow. Then we also have a paper of our user use PICS to search the team stock data. Similar, they have a target peptide list, and we analyze the data in our peptidome workflow. We can also report all of these target peptides. And then there is an application from the biopharma. In genetics, they use the PICS in their neoantigen discovery platform to help them to find some potential targets for the farm. And also in the research from Germany, they use the PICS to help develop the mRNA vaccines based on the immunopeptide data. And from academia, there is a new paper from the research from the US, from the Wisconsin Medicine. They use the PICS DeepNovo sequence. Based on this one, they develop their own algorithm to research on the neuropeptide and peptidomes. And here is some papers from our BSI describing our PICS online platform and the DeepNovo algorithm. So if you're interested, you can follow these papers. And here is some summary. So basically, the DeepNovo Peptidome workflow is a complete workflow for the peptide search. And the result is FDR controlled to increase identification confidence. We merged some deep learning approaches to predict MS2 spectra, the retention time, and the CCS to increase the sensitivity and the accuracy. And also, if we want to get a more accurate results, we give you the remote filter. You can filter on the CAA, on the MS2 correlations to get a more accurate result. Above all, our workflow is a sensitive and accurate tools for the peptide research. And before go to the demo show, I still want to show one paper from our users. They use the PICS-DB search to access the spliced peptides. So based on their paper, they find that our performance of PICS-DB is performs well, since we have the target decoy, fusion target decoy strategies to do the DB search to make sure the result is accurate. And in fact, our peptidome workflow use the same strategies. So that means we are always on the way to be better solutions. Okay, now we can go to the software to see how our software works. Okay, so we can go to the PICS online first. Can you see my screen?
Yeah, looks good.
Okay, so this is the peptide, the summary page of the peptidome result. So we can show how to use it. So we can go back. After you upload the data, you can just select the DeepNovo Peptidome workflow, and then go to the search page. You just include the search parameters here, and select the enzyme. Usually we just use the num, and then set the ppm. After that, you set you input the FASTA database, and you set the final FDR control for the result. Then you just click submit to submit the search. When it's finished, so you will have 2 pages. So in the PICS 11, we offer our result is a tree structures. So usually we'll have one summary page, and then it's the peptide page for more detailed results. In the summary page, you will see the summary statistics for each sample and for all samples based on some attributes in the MS1, MS2, and the peptide. And then you will get a FDR curve. So based on the FDR curve, you can decide which FDR may be the most suitable. And here is the statistics of the result. You can find how many peptides are from the DB search, from the spider search, and from the DeepNovo, and the final cutoff for the -10 log P. And here is the M over Z shift. You can see if the setting parameters is suitable. And here is the delta RT. Since this data is generated on the QEHFX, so it doesn't contain unweighted information. And the final we have a figure here is the delta RT across all the sample. So that means we know that we can notice that only in the start of the gradient, maybe the LC is not stable yet, so the delta T is long. And in the middle of the gradient, the delta T is very small, so the data set is very good. And then we can go to the peptide table. So in the peptide table, the first thing that now we have the peptide filter here. When you click here, you can use the peptide filter to filter the result. Usually we will recommend to use the CA here, and then it's the MS2 correlation here, and the delta T. And also in here, you can select filter all the peptides, or just filter the DB peptides, or filter the DeepNovo peptide. And in the result table, we have 2 views. By default, you will see the mirror plot here to see the measured MS2 spectra and the predict spectra. You can also shift to the classical PSM view to see here to check if the intensity is high enough. And this is on the peak. This is the DDA data. And also here is the DRA data. So in the DRA data, we will do the spectral library search first, and then the DB search, and then it's the de novo search is a complete workflow. In the DB search is the final result we use to compare. And based on the DB result, we can check the M over Z shift, and also check the regression of the IRT peptides to see if our RT predict model is good. And this is the same. We find that only in the last 10 minutes of the gradient, since sometimes we watch the column, so it's common that the delta is higher here. And for the peptides, it's the same. In the peptide table, we can have the spectra of the predict of the method and the predict. And over here, we can also give you the illusion profile. And then we will introduce the show the workflow in the Pix Studio. Since now Pix Studio, the most of the parts they are similar, or they are exactly the same in Studio and online. So in here, we just show some difference between the Pix Online and the Studio. So the summary page is exactly the same. We use the summary tables, we use some figures to show the results. And then in the peptide table, there is one unique features in the Pix Studio. That here in the Pix Studio, we have the feature view. So besides this PSM view or the mirror plot view here, we have the feature view to help the user to check if this peptide is good, something like this is a feature, then we can say this is the isolation window, and this blue plot means that these peptides have a good matched in the peptide search. And in the DIA workflow is the same. We also have the summary page, give you the figures and the summary of the results. And then it's give you the peptide table. And also in the Pix Studio, we offer the feature view here. You can check the feature to see if you are precursor or if you are fragment ion XYZ is good. That's all of our introduction. And if you have the questions, you can connect ourselves and follow our social media.